Okay, thank you, and happy birthday, Sriram, and also buon compleanno, because Sriram does speak some Italian, or as he says, he pretends to. Prego. Also, thank you for guiding us for so many years through the wonderful world of hydrodynamics, and for 3 very inspiring lectures earlier this week. One of the traits, I think I can say that one of the traits that we all love in Sriram is his unbounded enthusiasm and curiosity for all things new. And so I wanted to demonstrate that by showing you a little video clip that was taken 11 years ago, almost to the day, in November, when he visited us in Syracuse. And so here we go. Let me see, I haven't even turned this on yet. It was studying pattern formation at that time, so that was the Anyway, I'm not going to tell you about snow angels. Instead, what I want to talk about today is some work on how we can control topological defects in a couple of settings, in sort of active liquid crystals. And this is work that was done mainly by Suraj Shankar, who is here, by a wonderful undergraduate, Luca Scharrer, who's now a graduate student at the University of Chicago, and Mark Bowick. And if I make it, how perhaps Hydra or other living organisms might control a topological defect through interaction between nematic texture and biochemical signaling. And that's work that was done by student Zhihang Wang and postdoc at KITP, Fridtjof Browns. So we've heard about, well, this audience probably everybody knows about passive nematic liquid crystals, fluids of Rodman molecule that can order go from isotropic to a nematic state. And of course, the nematic state is characterized by an order, transition is characterized by an order parameter, the Q tensor, with the magnitude that is sort of the largest eigenvalue, controls the magnitude of the order, and the director, which has this n minus n symmetry, which dictates the properties of the material. Active liquid crystal, or active nematic liquid crystal specifically, are fluids of elongated agents that exert force dipoles on their environment. And as we were reminded by Zvonimir and others earlier in the week, it was shown by Sriram and Aditi Simha back in 2002 that such active nematics are intrinsically unstable, at least in bulk. And a few years later, actually 10 years later, in 2012, Zvonimir essentially developed this system, a suspension of microtubule cross-linked by kinase motor protein at the oil fluid interface, which has become sort of the paradigmatic example of an active nematic. And the key characteristics are these are self-sustained spatiotemporal chaotic flows and the presence of topological defects, the plus and minus 1F disclination, minus 1F, and the plus 1F, which become motile. Now, to be fair, however, and you saw this in one Sriram's earlier lecture, another realization of an active nematic actually is the work that involved actually Sriram. It was back in 2007, was this vibrated monolayer, vertically vibrated monolayer of granular rod that also exhibits, this is a dry system, there's no fluid here, but it exhibits a very similar type of chaotic dynamics. And furthermore, well hidden in the supplementary material, there was the prediction that the topological defects that can be mapped out here can become motile. And Sriram himself explained, I believe on Monday or Tuesday, I'm not sure now, where this comes around from this essentially curvature due current. Now, I'm actually interested in, I will be talking about active nematic fluid, the case where flow is important. And the mechanism, we understand it for this defect motility, we understand it a little bit differently, but really we should recognize that all this work on motile topological defects was really started with this paper. So to describe the atinematic liquid crystal, we do it hydrodynamics. That's the way Sriram would do it. He would write the equations down on the basis of pure thought. Some of us actually try to derive them from microscopics, but here they are. The fluid is assumed to be incompressible, so we just have equations for the order parameter and the flow velocity. The order parameter relaxes through free energy, and it can be rotated by shear flows and by vorticity. And we also have a stiffness due to elasticity. The system is essentially overdamped. We are in the Stokes limit, so we just write down the Stokes equation that balances friction with pressure gradient, viscous stresses, elastic stresses, and the active stress, which also was first proposed by Sriram back in 2002, which describes the density of, I might have a wrong, do I have a wrong sign? Or something, I don't know. Describe essentially the density of force dipoles exerted by these microtubules, and is proportional to the Q tensor to a quantity that we call the active stress. Now, I called it alpha. Alpha is minus zeta delta mu. And when we were writing our 2013 review, we had a lot of discussion whether it should be alpha minus zeta delta mu. I had to give up. I think we call it minus zeta delta mu in the review, but I'm back to alpha here. So extensile is negative alpha, and it's the opposite convention. And anyway, we're here talking about extensile system in this case. So, and these hydrodynamic equations do reproduce a lot of the dynamics quantitatively that is observed in atinematic liquid crystals. In particular, you can actually understand quite simply the origin for the motility of the +1F defect. The active stress acts like a source for flow. So you can actually put in the texture corresponding to either a +1F or a -1F defect and calculate the flow that that defect will generate. And for the +1F, which is not symmetric, the flow is these two vortices, and therefore is non-zero at the core, and the defect rides along essentially with the flow it itself generates like an active polar particle. And the direction of motion actually depends on the nature of the active stresses. -1F, the flow is even more complex, but is zero at the core by symmetry, and so the -1F is sort of a passive particle, let's say. Now, this is all something we did quite a long time ago, but what I want to tell you today about is the fact that, so there is actually essentially a one-to-one correspondence between the dynamics and statistics of the topological defects and the statistics of the active flow is characterized, for instance, by the distribution of flow vortices. And the defects can really be thought of as localized excitations that drive the dynamics in these active liquid crystals. So the question we've been addressing is, can we sort of look at the inverse problem, actually design activity patterns that will control the defects and therefore allow us to generate desired flows, something that could be used in the microfluidic design, or maybe biology uses something of this type. And there has been one of the motivation for this comes from the fact that it's actually possible to control the spatial temporal distribution of activity, for instance, through optogenetically modified motor protein, as these are experiments by Matt Thompson at Caltech. He has a microtubule kinase in suspension, but the kinases are light sensitive. And in this case, the microtubule assemble into an aster, and the yellow dot is sort of a column of light that is moved around actually by moving the table underneath. And the aster, in this case a +1 defect, tracks the light provided you don't move too quickly. So we want to learn how to do, to develop a formalism for something like this. And we can do it. So the goal again is constructing the profile of active stresses or activity that will give you a specific configuration of defects. And we do this by exploiting essentially the linearity of the Stokes equation and using symmetry first of all, the linearity allows us to use an additive approach and using symmetry to characterize the type of flows that they can arise from a given distribution of activity. So this is the Stokes equation again, written in a slightly different way. Now alpha is under gradient because we are looking at possibility of spatially and temporally varying activity. And if we solve this equation, again, using this as the source for the flow, the defect velocities can then be defined as the essentially angular average of the velocity and their vorticity at the core of the defect. Now, what we do is, so I want to notice a couple of things. So the flow structure clearly is dictated by the combined symmetry of the defect texture and the activity field. The defects, well, there are +1f and -1f, but it's actually more useful, instead of using their topological charge to characterize them, to use what we call a defect symmetry number, which is 1 for the +1f, which is a vector, essentially, and 3 for the -1f, which is a triad. Similarly, the activity profile, we're in 2 dimensions, so we can actually decompose it in polar components of defined rotational symmetry in the following way. Now, when we combine these 2 things, well, we put this in here, and we expand our velocity in polar coordinates, and we solve. And essentially what we find at the end is a sort of a simple rule for determining what kind of behavior that the given profile of activity gives for the defects. Specifically, what we find, remember, S is the defect symmetry number, and K is the polar component for the activity, the spectrum of the activity. So everything is controlled by this difference delta S. And if delta S is in magnitude equal 1, there will be the defect will have a non-zero velocity. If delta S is in magnitude equal to 0, it will have a non-zero vorticity. And all other values of delta S leave the defects unaffected. So for instance, constant activity profile, well, actually, I want to show the other slide. A constant activity profile will, as we know, get the +1f to move, but leaves the -1f unaffected. A linear activity profile, k equal 1, causes the +1f to rotate, but does nothing again to the -1f. But a parabolic activity profile can actually render the -1f self-propelled too, and also, sorry, adds to the propulsion of the +1f and also renders the -1f self-propelled as well. So what can we do with this? Well, we can design what we call a topological trap that allows us to move around defects. In particular, for instance, for the -1f, this is a -1f, we can do a little spherical region, circular region of activity. And this is a video I'm going to show you in a moment. Here you show the vorticity, here is the speed. And you see the defect moving as we move our circular region of activity in a precise trajectory. And this works as long as you don't move it too quickly, and too quickly means essentially at speed less than or comparable to the speed of the defect itself. And you can play around a little more with this kind of stuff, and you can actually generate a pair two pairs of defects and move them around and braid them around each other in trajectories. And what is kind of interesting about this to me is that actually the braiding of defect trajectory has been measured experimentally by the group of Linda Hurst a few years ago. And what they showed is that by measuring essentially the so-called topological entropy associated with the braiding of the defect trajectory, they obtain the same information that one can get from measuring Lyapunov exponents associated with fluid mixing. So again, demonstrating a one-to-one correspondence between defect dynamics and flows in these kind of systems. Okay, so we can move around defects one at a time. What about having many defects? So what you can now what we've done here, these are actually numerical results obtained by simulating the full hydrodynamic equations. And what there is a system which has a strip of activity with a profile as shown here. And what is plotted down here is the charge density of plus defects minus density of minus defects. Now, if there is a shallow activity gradient, what you find is that the minus 1f defect, the plus 1f defect can actually sort of escape from the active region and eventually enters the passive region. Of course, it loses its motility and it stops. These blue arrows don't indicate the defect velocity, they indicate the defect polarization that points against the tail of the defect. And conversely, sharp activity gradients act sort of like walls. Oh, and I should have said that, and down here you see that the result is that there is an excess of positive topological charge sort of just outside the strip, and an excess of negative topological charge inside. Sharp activity gradient really acts sort of like walls. The defect really cannot escape. And in fact, the charge distribution is flipped. There is actually an excess of negative charge close to the boundary of the active strip and the positive charge inside. And actually, you can understand this from the forces that defects feel in the presence of activity gradients. And so shallow interfaces are leaky, sharp interfaces act like walls. You can quantify this by measuring the topological dipole moment, the difference between plus and minus charge integrated over half a strip. And that quantity is plotted here as a function of the activity gradient. And what this shows is what we saw in the previous figures, essentially that the sign of this dipole flips. As you increase the activity gradient, you go from having the plus defects on the outside of the strip to having the minus defect inside the strip. So you have sort of like a topological capacitor that you can actually flip the sign here. And we actually have constructed some time ago a hydrodynamic theory of the defect themselves. This theory can actually predict this dipole flip, which, and that is given by this red line over here. So in a sense, you can say that activity gradients act like an electric field that can separate defects of opposite topological charge, and you can imagine using this for something. Okay. The other thing you can do, and this was really, I think this was Luca's idea, wasn't it, I think? The other thing you can do is actually take your strip and move it along at a constant speed. And I'm going to show you, these are 3 videos, but let me tell you first what I'm going to plot here. So there's going to be, this is the active strip, and it will be moving at some speed v. So what is plotted here is the net polarization of the plus 1f defect, and the number of defects that leak out of the strip as a function of the velocity of the strip over the typical velocity of the defects. Now, if the strip is moved very slowly, essentially nothing much happens. The defects are able to follow along adiabatically, and your turbulent strip keeps moving along in the system. When the strip is moved very fast, essentially, it kind of swipes along and cleans up the system. The defects are annihilated, and you end up with a system without any topological defects. At intermediate, when V is comparable to V naught, what happens is that it's sort of an optimal state where the number of defects that leak out of the strip is minimal, and actually the plus 1f defect acquire a mean polarization that is the system really orders. They acquire a mean polarization in the direction in which you're moving the strip. Okay, now I also wanted to just very briefly tell you about something we have done on topological defects in living matter. We have heard from Masaki Sano about the possible, about the fact that it has become in many more biological systems, one has found evidence for topological defects and possibly even a biological role for topological defects. And some images are shown here. Now, one thing, I don't know if Sri Ram has seen this before. If not, this is kind of my birthday present. Now, topological defects in living systems have been known for a long time. This is 1967 paper by T.R. Ellsdale, who worked at Western General Hospital in Edinburgh, Scotland, and looked at fibroblasts as they multiply and start becoming denser and fill the plane. And what is really interesting is the text here. So he noticed that eventually these fibroblasts order a line in patches. And it says, so the cells are ordered within a group, but the group themselves are disposed randomly. The groups occupy nearby all the area of a culture, and the narrow interstices between them will be termed frontiers. He didn't call them, he didn't have the term topological defect yet, he called them frontiers, but the notion of looking at topological defects in living systems goes back quite a long time. That's more a curiosity than anything. What we have done with Fridtjof and Ji Heng is kind of a very simple starting calculation to try to understand the role of topological defects in Hydra. There's been a beautiful work by the group of Kinneret Keren in Technion on these. So Hydra is a polyp, freshwater polyp, that can regenerate itself from a fragment. If you cut out a fragment of Hydra, it first falls into spheroid and is rather disordered. Then the sort of muscle fibers, these are really supracellular actin fibers on the surface of the body, exhibit nematic order, and then eventually the whole thing evolves and develops a mouth appear, a foot down here, and tentacles, and so on. And what has been pointed out by Kinneret Keren is that there is a correlation between the location of defects and the specific morphogenetic processes. For instance, the future head coincides with a first of all, nematic order on a spheres requires a total charge +2, so either 2 + 1 or 4 + 1f, or a +1 and 2 + 1f. And so what she noted is that future head coincides with a +1 defect, and the future foot coincides with 2 - 1f defect face to face with each other. And notice defects sort of repel like topological charges, so there has to be some process that forces them to be in this configuration. Now, the development of Hydra obviously is quite complex, and it's really determined by interplay of mechanics, geometry, and biochemical signaling. As Hydra evolves, first the sphere elongates into an ellipsoid, and so here we have a coupling between geometry and orientational order, which is at play. And one thing that is established experimentally is that there is an expression of a morphogen, Wnt, which is known as head morphogen, and that determines the body axis, so this will be where the head will actually form. So what we try to do is a very simple model to understand how the texture can be controlled by biochemical signaling. We simply describe the system in terms of a director field and a prescribed morphogen profile. And we're a free energy that consists of a term, the usual nematic stiffness, so if you like, is a term that tends to align fibers with their neighbors, and an alignment with the prescribed gradient of the Wnt or the morphogen. Now, and then we just do relaxational dynamics, minimize this free energy, and look at the configuration. In a planar geometry, the alignment with the Wnt gradient can unbind the pair of defects and drive the merger of two +1f defects. And you can construct a phase diagram in the plane of the strength of the coupling beta and the range of this profile. And you find that you can have no defects, you can have a +1 and 2 - 1f, or you can have this other configuration. But what is interesting here, okay, I am out of time, let me just show here, is that you can actually show by looking at the defect configurations on a given three-dimensional structure that sort of resembles vaguely the body of Hydra, that having a prescribed concentration of morphogen at the head drives a +1 defect of the head and drives a pair of just opposing +1f at the foot. Here, the +1 is driven by the coupling to the morphogen gradient, and here, the configuration of the 2 +1f is really driven by geometry. Furthermore, the work in the plane says that another spot of morphogen here can actually drive defect unbinding, which will then possibly lead to the generation of the tentacle or the budding. So this very minimal model captures the observed defect organization. Of course, what is missing is the feedback. We showed that morphogen and curvature can promote fiber organization, but you'd like to understand how those actually feed back onto controlling both the biochemical signaling and the shape of the organism. In this case, both are fixed. So with that, I have a summary, but let me just show you the next slide. I just wanted to thank Sriram again for 40 or more years that we've known each other for a long time, of great science and friendship, and looking forward to many more. And here are a few examples of Sriram doing science, studying phase separation, teaching François Brochard how to snowshoe, telling us what to do about the icicles in our house, and so on. And with that, I thank you.
Jay. Christina, this is just a comment. The paper by Elstale that you mentioned about, so it turns out there's a paper by Roger Penrose in the Annals of Human Genetics, which actually refers this paper and talks about stresses and defects.
Okay. I mean, I know about the work of, I think it was Oliver Penrose on the defects in the hand, but this is Roger Penrose. I say, okay, well, you should send it to me.
I had a question about the boundary of those regions with different alpha, right? How, is there a kind of a stress continuity there, or how does Q evolve as you kind of go through the boundary? Is there a jump in?
You mean in the strip, or?
Yeah, the strip.
Actually,
or the circle.
both cases, essentially we take, or in the circles, we actually take a hyperbolic tangent profile with a width which is small compared to the size of the. So you were asking whether there is so the activity is changing continuously. And
oh, I see. Okay, and so Q.
Yeah.
I see. There's
so,
no jump.
But you know, you might not have noticed because I went a little bit quickly. But the texture outside is affected. So the case where the gradient is shallow and the +1f leak outside, the outside, the nematic order is horizontal. In the other case, is actually vertical. So it actually affects also the nematic order outside. Yeah.
Hello. Yeah, the first one is great. So, so alpha depends on concentration. The alpha.
No, we didn't assume alpha to depend on concentration here.
No, in general, one could assume it.
Oh, yes, yes.
So I was just wondering, you know, for the sake, if I tried to get 2 plus halves to come together by just, you know, tuning alpha, is that possible? Can I create a concentration profile that'll force 2 plus half?
I assume you could, but I don't know. Suraj, this is personal. Suraj, he knows. I don't know. I think you could. By the way, we have shown some time ago by using the equations that we derive for defects as particles that actually with the right activity, that kind of, for an extensile system, that structure, the 2 plus 1f like that, can actually be stable.
I see.
I think, are you asking about the concentration of the morphogen in the second?
No, I think he's asking about the first.
First part.
So, well, he's asking whether one can design a profile of alpha that will, and I think so. I mean, I don't know exactly what that will be right now. Maybe Suraj knows what that will be off the top of his head. I don't, but you certainly could.
Yeah, one should be able to.
So that's the point.
Yeah.
Once you understand the flow generated in terms of this polar harmonic and so on, you can design any configuration you like, I think.
Okay, great, thanks.
I have there's a question there. But I'll just ask quickly, in the case of Hydra, is the claim that these defects happen to be co-localized with the head and the feet, or that they are causal in triggering
well, that's, I don't think, so the claim, the experimental claim is that the head, say, happens at the +1 defect. Now, and this is driven, and I think people would think that what drives the texture there, I mean, this wind, this morphogen really is what plays a role in driving that. Maybe Jack has something to say about that.
Do you want to respond to this, or you have another question? There's a question there. A comment, okay.
Then it goes first.
Please go ahead. Okay.
No, there is a very simple argument for explaining why a +1 defect could open a mouth.
Oh, well, of course.
It's very simple.
Because it's got
we proposed that with Tom Lebensky in 80 or something like that. Because you relaxed the energy by opening a hole. It's very naive, very simple.
Yes, but it's also true that the +1 defect means that the core is disordered and different, and so the disorder could be a hole, indeed. I mean, the core could be a hole. Okay, so you say that really the stress. Okay, all right.
In passive pneumatics, we see both the ±half and +1 defects. But in most of the active pneumatic, I see in the experiment and simulation that the majority of them are half defects.
Yeah.
So is there any specific reason why we don't see those, or even fractional defects, which are also seen in passive pneumatics but not in?
The only thing I can say, I guess, in active pneumatic, for some reason, +1 decay very quickly in this motile system to 2 ± 1F. I mean, I don't know. I know you're right. I noticed this myself. I don't know if Zvonimir has a better answer. See, I just call on people to answer my questions. No. Okay.
So I can comment briefly on that.
That's a good point. And,
what's typically seen in simulations is that the integer strength defects always break apart.
Yeah.
So if you do simulation work, they're always ±1/2. In the experiment, you stabilize them with some kind of impurities or with surface anchoring conditions.
So in the experiment?
In the experiments, you can stabilize the higher charge defects either with some kind of impurities that give pinning sites or with surface anchoring conditions.
But without something like patterning or...
So this is one of the main reasons
of course you can do that.
Experiment.
But without something like that, I think in the experiment also, the difference is that in the simulations, you can actually see the +1F decay in the the +1 decay in 2 +1F. I don't think you can see that in the experiment. You just see the ± 1F. I think.
Okay, so thank you, Cristina. Thank you all the speakers for the session. So.